Welcome to the Hawaii Book and Music Festival, the wellness track. We've got a dynamic panel today, 3 panelists, to discuss gene splicing, gene therapy, gene editing. We have Dr. Jess, and all 3 of our panelists are PhDs, PhD professors. We have Dr. Owens, Jesse Owens, who will present first. He's a local boy and has done studies at JABSOM, looking at transposons for his PhD. He's published quite extensively, and now has his own independent lab where he's working to optimize novel gene therapy. Also with us tonight is David Segal, Professor Segal, whose focus is also with gene therapy, primarily on neurologic disorders. And our third panelist is Johann Urschitz, who is looking at optimizing the health of the placenta and minimizing disease around placental dysfunction, fetal growth, and adult disease. We'll start off with Professor Owens, who has a slide presentation, and then move on. There will be roughly 15 minutes for questions at the end. Professor Owens, you're on mute. Yeah.
Thanks. Just get our slideshow going here. Our pointer. Okay, well, welcome to our panel. Our title is Breakthrough for Safer Gene Editing. And I was going to go through a quick presentation of some of my work to kind of set the stage for the kind of topics that we're going to be bringing up here. So I work on gene editing technologies, which basically means that we try to bring in genes to replace dysfunctional genes. So if you've got a mutation where some of your bases are wrong, your gene might not work, and that might cause a disease. And what we do is we go in there and fix the actual sequence that makes the gene and makes the protein, and try to treat your disease. But my lab is especially focused on just the methods. So the kind of what we hope is that the tools that we develop in our lab will later be taken beyond the lab into the clinic, into different model organisms like mice or dogs, and then once they show that's safe enough, even into the clinic for people. So I'm like at the very beginning of the treatment side of things. So just a quick overview of the kind of different tools that are out there in the toolbox. In the last few years, it's been very exciting. All these new developments have come up with different ways to modify the genome. And I'm not going to go through all the details, but just to give you an idea of what we can do now, there's this thing we call base editing, which is really neat. What this does is it allows you to change a single base for another base. So there's different kinds of corrections that can happen. You can say, okay, I've got a broken gene. Why is it broken? Well, maybe it has an A where a G is supposed to be. Well, with base editing, you can go in there and swap it for the correct base. This is a pretty recent discovery, and it's extremely efficient. It's really nice. You've probably heard of CRISPR before, and I'm going to quickly go through what CRISPR, a very broad description of CRISPR. But basically, what CRISPR is really good at is causing deletions, and this has been hugely important for a lot of the studies in the labs where we're trying to knock genes out. Because the best way to tell whether or not a gene is important or what does a gene do is you knock it out in a mouse, And then you look at the mouse and you say, okay, well, what's different about this mouse than a regular mouse or than a wild type mouse? And you know, if the mouse has heart problems, well, then maybe your gene is involved in something to do with the heart. And so this has been been really nice to be able to efficiently knock out genes intentionally. So CRISPR is really great for making things broken. We have this new therapy called prime editing now. And what this does is it allows you to bring in small sequences without using this method that I'm going to describe called homology directed repair. And that we'll come back to that in a second. We also can use the same CRISPR system that we use to knock things out to bring things in using homology directed repair. And I'll quickly go through that. We also have this thing called CRISPR Effectors. So these are things that we don't actually change the genome, we modify it in some way. And this is the stuff that panelist David Segal has really been killing it with, where we change the epigenome, which is the kind of the things around the sequence. We don't actually change a G for a T or anything like that. It's modifying the expression of genes. You don't actually have to cut the genome. You don't have to mess with it. So that's a whole field that's been really taking off. And the stuff that I work on is targetable integrating enzymes. So these are enzymes that will actually take a big piece of DNA and stick it in the genome. And so you could use that to replace like a whole broken gene or stick in whatever you want into the genome. And one of the key things about why what I do is different than HDR is that we don't require homology directed repair. So real quick, I'll try to go through what am I talking about with this. So with CRISPR, the basic idea is we have a small piece of RNA and we have a protein called Cas9. Now the RNA binds to a specific sequence in the genome. So if we want to cut a sequence in our genome, we make a RNA that is the same base pairing as the sequence that we want to cut. So feel free to stop me and ask questions if you have questions. But basically, if let's say we're trying to chop up GTTA or whatever, we can actually just put those same base pairs on the RNA. And what happens is the RNA will bind to our Cas9 protein and will actually locate that sequence in the genome, and Cas9 will cut that sequence. Now, that is all that CRISPR does. It just causes a cut in the genome at whatever sequence that you want. Now, that's really exciting because what happens next is often the genome will have to correct this hole. It's got to fill in the hole. It's made a mistake. And it doesn't do a perfect job when it does that. When it's like, oh, shoot, we've got these two strands that are broken. We need to do something about this. It sticks them back together. And when it does that, it often makes mistakes using a process called non-homologous end joining, where it just sticks it back together any way it can. Sometimes it'll insert a little extra sequence. Imagine when you're trying to stick like two 2x4s together, you got to put a little bit of glue in the middle to like make them stick. Well, that's what it does is it basically adds a little bit of sequence there to make them stick. And in that process has disrupted it because it's a new sequence. It's mutated that region. And so we can use this to intentionally make mistakes in the human genome. Or what we can do is use homology directed repair where we bring in a sequence into the genome now. And so what we do is we have our gene of interest that we want to bring in. And this sequence here has similar sequences to the nearby sequences. And we trick the cell into thinking that it's going to correct the mistake by using what we've brought in. So basically what happens is the cell says, oh, we've got a cut. Well, I want to make sure I put it back together properly. So I'm going to use this sequence as a template to make sure I do it right. And when it does that, it accidentally brings in your gene of interest right at that site. Now, which is really cool. So now we've got our sequence, we've delivered it into the cell, but the cell had to do that. So the CRISPR doesn't do that. The cell does that. And so we're limited to how well the cell can do that. And it turns out that the cell's not really that great at accidentally sticking in genes of interest at the cut sites, although it can do it. It especially does this well in dividing cells, but it's not that great at doing it in the non-dividing cells. So in my lab, we mostly work with dividing cells because we work on tissue culture. And so we've got like a dish, and we've got cells that are like stem cells, pretty much. They can divide forever. And so this isn't a problem. We can use CRISPR, we can stick things in using HDR, everybody's happy. But when we're talking about a therapy for a person, we're looking at the cells of our body. Most of the body does not divide very often. You have a little bit of cell division in your hair, your stomach, your stomach lining. But in general, most of your cells are non-dividing. Your stem cells are dividing, but the vast majority are non-dividing. That's why you can take chemotherapy and survive, because chemotherapy kills off all the dividing cells. So this is a problem because CRISPR, while it's really good at cutting and making mistakes in our dividing and non-dividing cells, so knocking something out. But when we try to stick something in, we can do it in the dividing cells, but we have a hard time doing it in the non-dividing cells. And so something that I've really focused on is this need. So we have a lack in the ability to bring in sequences into the non-dividing cells. And so that's where I come in. What I'm going to do is, instead of just allowing the cell to kind of figure it out, we're going to actively take the gene that we want to bring in and put it into the genome. So it's more of an active process. Instead of just cutting it and say, oh, I hope the cell figures out this, cleans up this mess, we're gonna actually actively insert it. And so the way we do this is using this thing called a Transpose Ace. So Transpose Ace is a very fascinating protein that is a part of a transposon. So Transpose Ace is a protein. Transposon is the sequence that gets moved around. Okay, and so this is what we call selfish DNA. It's DNA that actually makes a protein that is able to cut itself out, the DNA out, and hop it around. It was just when the person, Barbara McClintock, discovered this, everybody thought she was crazy, but it turns out she was not crazy, and she got the Nobel Prize. So what we've done is we've borrowed this, I guess you could call it a technology, in order to deliver genes into places. Because what we're trying to do is we're trying to hop a gene in, or a sequence into the genome, and this thing naturally does that. And so what we've done is we've kind of borrowed that ability. So you can bring in your Transpose Ace either as a protein, or as an RNA that gets turned into a protein, or as a DNA that gets turned into a protein. And then that protein, the Transpose Ace, will cut out the transposon and then send it into the human genome permanently. And so that's how we get things in. And this is basically what I've been working on. And now I would like to go kind of quickly through the benefits of this type of therapy versus the other therapies that are out there. So the way that people generally are delivering DNA and genes into people right now is by using viruses, because, as you know, viruses already do this. And so people have borrowed the viruses in order to deliver their genes. But there's some drawbacks to the viral approaches that people are using. One is that they can't carry large genes or multiple genes. They have a limited size, a cargo size that you can use. Developing them is pretty expensive. And a lot of people, because these are viruses, we build an immune response to them. A lot of people have a pre-existing immunity. So some of the popular ones you actually can't give to people because they've already been exposed in the past, and they've built immune antibodies against them. So that's a big problem, actually. Something like 40 to 60% of people actually are not able to receive some of the more popular viral approaches out there. So that this limited cargo size is a big problem. And having large insertions would be really nice. So some therapies require large genes. The promoter, which is the part that controls the gene, can be large. Sometimes genes require alternative splicing and introns, which make the genes bigger. And also sometimes some of our more complex therapies might require multiple genes. Depending on the disease, as the diseases that we try to treat get more and more complex, we might need to bring in multiple genes, which are too big to fit on one virus. So it's important to be able to bring in big sequences. And like I mentioned, CRISPR can bring things in, although it requires that host cell proteins, the proteins of the cell. It doesn't do it on its own, which makes it inefficient in non-dividing cells and inefficient for large insertions. So again, our tool in the toolbox is bringing in big sequences safely, whereas some of the other tools you might be just like changing a single base pair. So the transposons that we work on are very efficient because they actually do the reaction of moving the DNA into the target DNA they don't need those host proteins. They're efficient in all different kinds of cells, and they don't just break open the DNA like I showed you in that first slide, and then to say, okay, figure out how to clean it up, right? So it protects it as it inserts. So kind of the goal of my lab is to figure out a way to get the transposons to insert at a very specific sequence. And so what that would allow us to do is insert our gene away from important things. So we can put our gene somewhere where it's not going to hurt anything. And we also want to make sure it goes to a place where our therapeutic gene will be active and expressed. Whereas some places in our human genome, if we put in the gene, they'll get silenced, and they won't be useful. So we have these kind of two things we've got to figure out. And so in order to accomplish this, what we can do is choose where we send it, and then now we know it's not going to break anything, and it'll be expressed nice and bright. So the concept here is we have our target sequence in the genome, and what we do is we make a DNA binding domain, which is a protein that binds to that sequence. And then we add a leash to our transpose ace. The transpose ace that I work on is called piggyback. And so now the piggyback transpose ace, the protein, has been localized nearby the sequence where we want it to insert. So had we not included this leash, it would just be running up and down, inserting wherever it wants. But now it's localized, it's stuck nearby the sequence. And so then what it does is it does what it does best. It inserts our gene of interest. And then after all the smoke clears, we've got our gene of interest sitting next to our target sequence. So we're basically like shooting at a target here. Whereas had we not done that, this gene of interest might have been inserted somewhere where we don't want it. Like maybe it could have interrupted a different gene, which can lead to cancer. So it's really essential that we target where our genes of interest go. And now just to give you some actual data from a paper that I threw in here that I published with Dave Segal, who's on the call, we actually isolated cells, and this is like a colony of cells that are human cells that we grow in the lab that actually had that targeted gene only 22 base pairs away from our target sequence. And this is a PCR, just like the PCR test they do with COVID, see whether or not COVID's there. Well, what this question was whether or not our insert was there. Did we actually send our therapeutic gene to where we wanted? And sure enough, we did. We got a band there. So we are capable of directing these inserts to the site that we want and away from other sites. And so what this is showing here is that we only inserted one transposon or one therapeutic gene, and we showed that it wasn't silenced. It stayed nice and it expressed for a long term. And this was the first demonstration that a transposon can be targeted to a single user-defined genomic location or a site in the human genome. So yeah, we're really happy about that one. Now, what we've kind of moved on to next is a way to kind of combine that CRISPR approach with the transposon approach that I just described. And so this is kind of cool. What we've done is we've taken kind of the parts of CRISPR that we like, but we left out the parts that we don't like. So usually CRISPR is able to cut the genome. Remember, I said it cuts the genome and we didn't really like that. Now what we're going to do is we're going to use our RNA to target whatever sequence we want, and it binds on to dead Cas9, and dead Cas9 is able to bind the genome, but it can't cut the genome. And now it kind of looks like our initial picture where Cas9 has brought the piggyback transposon to the site, and it can insert nearby. And so we've borrowed kind of the benefits of CRISPR to target our transposon. And so what we're now working on is this concept of making those insertions 100% necessary, 100% reliant on the Cas9. And so basically, this is like our model for the future here, is what we currently have is this ability to bring the piggyback transposon to the target sequence, and then we get the insertion nearby. But we also have off targets that could happen where the piggyback transposon, Transpose Ace, is able to insert without using the Cas9. So we get a mix of on target where we want the inserts to go, and off target where we don't want them to go. They could be bad places. So what we're doing now is we're mutating the transposon so that it uses the Cas9. And if it doesn't use the Cas9, it doesn't insert. And so that's kind of the focus of my lab right now. And without going into the nitty-gritty details, just showing you kind of like a picture of what this is actually what piggyback looks like. And what we've done is try to figure out what is it going to take to make this Transpose Ace rely on the Cas9 so that we only get on target and we never get off target. We kind of zooming in here and like mutating it so that it relies on the Cas9. So yeah, this is kind of the stuff that we're working on in my lab. So anyway, so kind of the point of this little short presentation was to give you a taste for the challenges out there, because we want on target, we don't want off target, and a little bit of detail about how I'm personally doing it in my lab. So moving on from there, I thought we would spice things up a little bit. And just really quick, I'd just like to give an acknowledgement to my lab and my funding and my past lab members and my friends who I collaborate with right here. I'm funded by a bunch of different funding streams. Anyways, so moving forward and to kind of spice things up a little bit, what I thought would be fun would be to bring in some gene therapy experts, Dave Segal and Johann Urschitz, who could help me discuss some of these hot topics, if you want to call them that. So the kind of the three discussion topics that I came up with are the dangers of gene therapy versus the cure. So like I said, and even in my therapy, I don't have my therapy working perfectly yet. The one that I'm designing still has off targets. And so an off target event might mean that the gene that you bring in disrupts a gene that you already have, which could be bad. It could kill you. So there are dangers to bringing genes in. However, we're trying to cure diseases, right? So the back and forth is, is it worth it? Is the risk worth it? The second topic is what would be possible with gene therapy in the next few years. In the last few years, we've seen this huge boom in what we were able to do. Most of those different tools in the toolbox that I just described have actually been developed in the last 5 years. So we're wondering what might come up next. And then the other question is germline gene therapy, which means what happens if you add a gene to the sperm or the egg, so that when you have a child, they already have that gene. Because as you know, from that point, once it's in the egg, every single time it divides, your gene will be there, and in every single cell of your billions of cells in your body will have that gene. And when your children have children, that gene will continue to your grandchildren. So it's in there forever. So germline gene therapy, if it's so safe, why is it still forbidden? If I'm successful and I get my therapy working really well, why can't we use it for germline gene therapy? So these are kind of things I thought would be interesting to talk about with Dave and Johann. And so maybe we'll switch over to Dr. Segal. Dave here, he can give you maybe a little quick description of what you're all about, and then maybe we can start discussing these topics.
Yeah, great. So thanks everyone for tuning in and letting us talk to you for a little while, and hope that we can make this a kind of interactive discussion. So certainly if you have any questions, let us know, speak up, put them in the chat, whatever you'd like to do. So my name is David Segal. I'm at UC Davis in Davis, California. And the work that we do in my lab is to try to develop therapies for neurologic diseases. So, to kind of pick up where Jesse left off, I mean, basically he was describing, kind of advanced methods for how we can actually go about very precisely changing genetic sequence in cells or regulating the expression of genes in cells in a very precise and controlled way. And as he was describing it, these methods have really advanced in recent years. So there's a lot of tools to be able to do this, and people are trying things in cells, and some of these are advancing to the clinic. It's nice now to be able to say that we live in a world where gene therapy is possible, that is actually happening. For most of my career, there was a lot of talk about gene therapy and, when was it coming? Oh, it's coming soon. It's right around the corner. Oh, we've had a setback. Oh, we've had a success, and always in the future. But now there are actually a handful of gene therapies that have been approved by the United States FDA, Food and Drug Administration. There's been some that have been approved by the European Medical Association, the EMA. And these are now treatments involving this kind of gene therapy that patients that suffer from these diseases are actually able to access. So we're living in a world now where these things are actually happening. It's no longer something that we're thinking about in the future. Now, there's not a lot of them right now, but there's more being approved all the time. I think the last one was approved just a few days ago, and the other one a few weeks before that. So we're going to be seeing a lot more of this. And as the field progresses, the technology is getting more refined and able to do more kinds of things. So some of these methods are, I guess, in the development of these gene therapies, the gene therapy that a doctor can use to treat a patient, kind of the very end of the process. But along that, there's like what we could call preclinical development, where we're still trying to work things out. We're working in cell culture, we're working with test animals, that kind of thing. And so that's all the preclinical work, trying to build up to making a therapy. And then there's the famous period that many people have heard about, the clinical trials, where things are actually tested in people. So when you hear the term, those are experiments that are going on in people, trying to see if this therapy is going to be safe in people and that it actually works in people. So those clinical trials could go on for several years, trying to get enough evidence that this process is both safe and efficacious. So those are the two critical words there. It has to be safe, and it should have a positive effect on the people that are being treated. So that can go on for a number of years, but then if there is enough information and the regulatory bodies like the FDA think that that's sufficient, then they'll say, okay, now it's been approved by us, and now a doctor can actually prescribe this thing. And so, like I say, there's a small handful that have actually gone through that whole process and are available. There's some more that are going into this clinical trial stage. And so some of the tools that Jesse was talking about, the things with CRISPR and all, there's several companies that have CRISPR therapeutics in clinical trials. So these are being used in people right now. And we're learning about how safe they are. We're learning about how effective they are, if they're working the same way that they did in the animal model, and if they look like they're going to be safe and do their job. And so a lot of the work that we do is in this kind of preclinical phase where we're trying to use the new tools and trying to see how well they can change the DNA or change the gene expression. But then, we're hoping to go into clinical trial with that. And from clinical trial, we hope to get all the way through to actually having approved therapies that a doctor can apply. So getting around to these dangers of gene therapy versus the cure. Certainly, this is a new kind of capability. And with all new capabilities and new technologies, there's a certain amount of concern, whether these are going to work, whether they're going to be safe. These kinds of questions come up. And I think in this discussion, it's important to consider that there's different kinds of classes of diseases, but there are many diseases right now for which there are no treatments. So, we could talk about diseases such as cancer. Cancer is a very big disease, right? Lots of people working on cancer, but still a lot of work needs to be done on cancer. So we've come a long way in treating cancers, but there's still a lot of cancers out there where we don't have effective treatments and still trying to work on that. And so many people are doing cancer research. Infectious disease is another huge area where we're trying to develop effective treatments and effective preventative treatments. We're all familiar with COVID now, more familiar than we would like to be. And actually, between cancer and infectious disease, those are some of the largest areas of therapeutics that are being developed, gene therapies or drug therapies alike. My laboratory works in this area of rare diseases, and rare diseases are diseases that affect less than 200,000 people in the United States. But there's about 7,000 of these rare diseases, and collectively, they affect more people than cancer and HIV/AIDS combined. Now, for almost 95% of these rare diseases, there are no treatments available at all. And one of the reasons that there are no treatments available is that these are genetic diseases. They're the kind of things that Jesse was talking about, where there's a gene that's mutated causing the disease. And sometimes that's really hard to treat with a drug therapy. But with these new tools that Jesse was talking about, we now have the possibility to try to treat these genetic diseases. And so, we're one of the labs that's trying to work on that. We are specialized, our lab is specialized in working on diseases in the brain. The brain poses many challenges to bring these types of therapies into the brain. But those create challenges, and we try to come up with creative ways to overcome those challenges. And this is a tremendous unmet medical need, because there's all these diseases affecting the brain, affecting other kinds of systems, and there's no treatments available. So
yeah, one of the things that's nice about these therapies is that you can put whatever sequence you want into the tool. So it's not like you have to redevelop your tool to switch up your gene. You can just change out the sequence and then choose a gene, a different disease.
Yeah. So I'm gonna turn it over to Dr. Urschitz now, but I just want to say that there are a lot of people out there that are desperately in need of treatment. And we always have to consider in a discussion about what are the risks of the therapy versus the risks of the disease. We're looking at people, they have a 100% chance of disease, right? They have a disease, usually very debilitating. And we have to weigh the risks of the treatment in that context, because we don't want perfect to be the enemy of the good. And we want to be able to bring treatments to people that desperately need these treatments, still keeping it safe but realizing that if we can do something to help them now, while their children are young and while they still have, you know, a chance to lead a better life, that's a strong consideration that drives our research.
You know, Professor Segal, could I just ask you, you know, you mentioned that you focus on the brain and neurologic disorders. Could you give us just sort of run us through one example of something that you've worked on, a certain neurologic disease that you've worked on, and where you're looking at the risks and benefits of therapy? Just a quick model.
Sure. So we've done a lot of work with a disease called Angelman Syndrome, which is a disease that probably most people on this call have never heard of. And that's good. That's good because it's rare, and it's good that it's rare because it's a terrible disease. People with Angelman Syndrome have a mutation or a similar problem with a gene in the brain. The gene's called UB3A. And people that have a mutation in this gene have severe cognitive problems. They never develop speech more than a few words. It's something that shows up in about the first year of life. And fortunately, it's not progressive in the sense that life gets worse and worse and worse, but they still have a lot of problems. They have characteristic movement problems. Some are able to walk. Some people will learn to walk when they're 13. Some will be wheelchair bound for the rest of their life. They often have seizures. Some of those are controlled with seizure medication. Some of them are not. And many other kind of cognitive problems as well. So they usually require lifelong care, and all that comes with that. But you know, we think about trying to either introduce or in some way activate this UB3A gene. And we're trying to do that with several different of these kind of molecular mechanisms like Jesse has described. And we don't have something in clinical trial yet, but we hope that within several years, we'll be able to be at a point where we can change the outcome of this disease for people that are affected.
Yeah, thank you. Thank you. So we've heard that, welcome to the Hawaii Book and Music Festival, the wellness track, looking at gene editing, mostly for therapeutics. We've heard from Professor Owens on his work, and Professor Segal focuses on neurologic disorders. Professor Urschitz, Johann Urschitz, works in the area of placental health and placental fetal interactions around and relationship to gene editing. Also, Professor Owens had kind of thrown down the gauntlet of three key questions that we wanted to look at, kind of the risks versus the benefits of therapy, you know, where the future is going. But one area that we haven't touched yet, Dr. Urschitz, is on kind of the issues of germline therapy. So I was hoping, in addition to speaking about your work, that you could define a germline and talk about sort of the ethical, legal, political, religious implications of germline therapy.
Sure. Good evening. So as Dr. Zunin mentioned, my name is Johann Urschitz. I work with fetal abnormalities due to things that go wrong with the placenta. And you know, like, while Jesse works on methods and Dave is much more advanced with like his, well, fortunately not his disease, but the disease that he's working on, I'm sort of like in between. So I'm pre-clinical trial. Some of the methods that I'm using that are gene therapy methods are I actually use to figure out what things or what genes don't work as they're supposed to work, so that I can then go in and do gene therapy on. So as I mentioned, it's all about the placenta. So a fetus can be too large and too big, too large or too small, excuse me. And I'm trying to figure out what exactly are the genetic line that it follows, why that happens. the problem is not just at birth when it's too small or too big, but those are too small or too big have a preposition to become overweight, develop diabetes, and have heart problems. It's called metabolic syndrome. Where I'm sort of like trying to do something in technology is, as Jesse mentioned, a lot of things rely on viruses, and I'm trying to develop or apply a method that has been developed to the placenta where I can change the gene expression within the placenta. So viruses are gene delivery machines. That is great, but they have disadvantages. So it would be nice if you had a method to deliver genes safer. And for me, the placenta is an ideal target because I'm not trying to influence the genes of the mother or the child. I'm trying to influence the genes of the placenta that are essential during development and determine, like health and disease of the acute during pregnancy. I mean, do you have like an early delivery, for example, and you are tiny? Or are you too big and you get stuck and you're lacking oxygen and have brain damage as a consequence, but also like later in life. So that's what I'm working on. So that's like an aspect where like I'm sort of like waiting for Jesse to develop his safe method that I can then actually say like, oh, I could use this for humans because I'm more what's happening and how can I deliver the method. So when it comes to germline gene editing, so germline gene editing basically means you're either editing an embryo, so usually a one-cell embryo, one-cell stage embryo, or you edit the oocyte, or the egg, or the sperm. The problem is, as Jesse mentioned, is that like this is not a sperm, and this is not a mutation that is temporary. So it's like it's there all the time and in all cells. And while many things have been tried out in animals, in model animals, model diseases, you can't transfer it one-to-one to a human. So this might work in mice or in monkeys or in dogs, but it may not necessarily work in humans. These things are much easier when you have a drug because you can play with a dose, for example. You can instantly cancel a trial if the side effects are too large. If you have a fetus develop through whatever to an adult life, that's nothing you can change anymore, really, other than potentially doing another gene therapy and knocking out what you've just created. So that's one of the problems. I think it's ethically and generally considered a no-no to go to germline editing, simply because I think it's too much of a I don't want to say insult, but like too much of a manipulation to be considered good. No, you didn't mention it. You mentioned it earlier. So there was a trial, or there was one pair of twins, I believe, were born that were germline gene modulated in China. It caused a huge brouhaha about it. The researcher who did it went to jail, not necessarily because he was gene line editing, because he was, I believe he was a PhD who was taking on tasks of a physician. That's what he got ultimately sentenced for. So, I mean, there is like certainly a number of challenges that are involved or that are around like germline editing. Ultimately, I believe it probably will happen at some point. There's always somebody who doesn't care what the ethic consensus is or the religious consensus. There's too many interests, and then like there's also like there's also, particularly in the US, there is there's like the bodily. Well, there's a couple of actually things in the Constitution, right, where you have where they basically lay out the reproductive autonomy, for example, in the Fourth Amendment, or the bodily integrity. So how do you define bodily integrity? Is germline, potentially is germline germline editing restoring bodily integrity? Yeah, we could maybe argue that. You could also argue that, like, hey, that's my body, you are my parents, you don't have to do anything, you're not allowed to do anything about it. That's not, I wouldn't allow you to do that. Again, then there's like the reproductive autonomy, which means like the parents can theoretically decide. So there's like a lot of ethical issues that have been, I mean, the closer we get to that, and the better the methods are, the more there is a need for it to lay a foundation of what's available, what's not available, what's doable, what is acceptable, so, and what's ethically okay for us to accept, essentially. I mean, this will be like a hot topic as I think as abortion, because ultimately this is about like how you perceive the bodily integrity versus let's say from the mother versus the child. So it will be, and this is just the US. That doesn't mean that like some other state has a completely different perception or a different way of looking at these issues.
Great. Yeah. So again, welcome to the Hawaii Book and Music Festival. This is the wellness track. And we're in the middle of a fascinating discussion on gene editing for therapy, among other things, from Professor Jesse Owens, from Professor David Segal, and just now from Professor Johann Urschitz, all three PhDs in research in this area. I think it was you, Professor Owens, that mentioned, or maybe it was you, Professor Segal, but that there were, in recent weeks, there were actually 2 gene therapies that were FDA approved. Can we talk a little bit about what is FDA approved now? How many therapies, what therapies? Because as we know, once something's FDA approved, it tends to make its way into being covered by insurance. So that kind of drives what we might have access to for our own care or children's care.
Do you want me to field that one?
Sure.
So the first gene therapy to be approved in the UK, I think, was in 2017. Glybera, actually, it was a little earlier than that, a few years earlier than that, but it was called Glybera. It was to treat a rare disease, and it cost about $1 million for treatment. And it was treating a very rare disease where they were only expecting to be able to treat about 10 patients per year. In fact, in part due to the high price and the way that reimbursements and decisions of cost effectiveness are done in Europe, they only treated 1 patient in its entirety. And at that point, the company felt they couldn't make money on it. And 2 years after it was approved, it was withdrawn from the market. The first gene therapy to be approved in the United States was something called Luxturna, where it's treating again a fairly rare eye disease, and was effective at doing that. The cost was about $450,000 per eye. So to treat both eyes, it was about $900,000. But that still remains a gene therapy that is being used. you mentioned reimbursement from insurance. That's become a very major issue for the gene therapies that have been approved, because many of them are expensive like this. And I think some insurance companies are, it's new, it's still early days, but I think a lot of insurance companies are apprehensive about paying that much money. This could be a whole discussion in itself about how these prices are set and what are the justifications, and what would be the cost of treating someone over their entire life, and how does that compare to a one-time treatment of, say, $1 million or so. But it's worth mentioning that a more recent FDA-approved gene therapy was something called Zolgensma. And this was a treatment for spinal muscular atrophy, another very debilitating, lethal disease. Zolgensma was first priced at about $2.1 million per treatment. There was another one that was approved a few weeks ago for $2.5 million, and I just read that the latest gene therapy was approved for $3 million, or it is priced at $3 million. So I guess in summary, the costs are very high, and I think there could be a lot of discussion around the implications of that in terms of reimbursement, in terms of accessibility and equity in who's going to be treated with these things and who can access it. But in terms of the diseases covered, a number of them are rare diseases that are chosen largely because they seemed, for various reasons, to have a high probability of success. So they're the first choices.
Yeah, I mean, we're seeing it throughout medicine, and right, the future has arrived. So we're talking about gene therapy here, but certainly the novel targeted therapies, the monoclonal antibodies, and so on and so forth for many cancers are really exploding in terms of their approval by FDA, and they're 500,000 to 1 million per year on average. The T-cell, cellular therapies for cancers and so on. And it's also interesting because in terms of cost, in the US, we tend to think that, we want to have and should be able to have access to anything that money can buy for healthcare. In Europe, it's driven by the national health services and, what's approved. So there can be a big difference. But I agree. I mean, it could be a whole separate discussion, and the pressure is mounting in terms of what science can do and how much it can cost. Other comments?
Maybe you can check if there's any questions.
Well, I'm watching the questions here.
Okay.
And I saw a couple flash up, but we're clear. We're clear on questions now. Are there any relatively common diseases that are near for which a gene therapy is near FDA approval?
Yeah, I think there's a few. One disease that I'm kind of connected to is high cholesterol. So there's people who have they're genetically predisposed to having high cholesterol, and so it's somewhat rare to have these diseases, and so that they're kind of the initial treatment group. But if that works and the gene therapies that we're trying to develop is able to treat these people, the idea would be to then make it available to the millions of people that have high cholesterol all throughout the country. So it's or the world. So there's a few of them out there. I don't know how close we are to getting that out into the just yet, but hopefully somewhat soon.
And it also may depend a little bit on what you call a gene therapy. So there's a cancer treatment I think you mentioned that goes by the name of CAR T therapy, and this is a very effective way of treating leukemias. And in order to do this cellular therapy, they have to introduce a new gene into those cells. So some people consider that to be a kind of a cell and gene therapy, and that is treating a common disease of cancer, leukemias in particular. So that could be another example.
Yes, yeah, so true. You know, Colin, there, I'm not seeing questions in the usual question and answer
It's in the
area, but there's
chat.
in the chat, yeah.
in the chat.
I think for the
Oh, okay.
yeah, for the costs, the companies that are developing the therapies, I think, are hoping that the insurance will pay for them, and that was the issue with it's called Glybera, right? The name of the that they ran into trouble because in that particular case, there was actually dietary ways of kind of not treating the disease but managing the disease. And so the insurance company said, well, you could just change your diet. You don't need this $1 million shot. And so they just didn't pay it. And that was one of their big problems. So it's up to the insurance company whether or not they agree that the therapy is worth the money. And so that's what Dave was saying is an issue.
Yeah, I mean, you know, it's really, when we talk about institution of healthcare insurance, you know, in the national health services and in the rest of the kind of modern world, they're just decisions made. Well, we cover this, we don't cover that. If you have this, we'll do this, but not that. But in the US, there's a lot of pressure. Once it's FDA approved and then it's Medicare approved, then insurances have a lot of pressure to follow. And what we found, you know, like there was that treatment for Hep C, right, for persistent Hep C, where it was 600,000. The R&D costs were far lower, but they said, look, you know, we're charging this because otherwise, you know, you'd have to have a liver transplant, and so we're saving you 200,000. You know, but what we see in American healthcare is that although insurance companies are under pressure to cover it, there's then just a lot of obstructive processes that clinicians have to go through to gain authorization. That's the bottleneck. So, but you know, I think this question, and there were a couple of chat questions about it. You know, that this is definitely a rapidly mounting issue around healthcare delivery and access.
I just want to mention, I mean, gene therapy at the moment is at the very cutting edge, literally, right? So if you find therapies for more common diseases and they are applied in the thousands or 100,000s, then I'm sure the price will go down as well. Obviously, that isn't the case. So, I mean, just if you can even consider COVID some kind of gene therapy, right? And because of their system, you can easily adapt the first COVID vaccine to a modified COVID vaccine by adding a modification that is better for Omicron, for example. And if like things like that happen, for example, Jesse develops or like has full clinical approval for his method, and the only thing, quote unquote, you have to do is change maybe the target or not even that, and then just the sequence that you put in, then you know, like costs will go drastically down. But you know, like drastically down can still mean hundreds of thousands instead of millions. But you know, like it's again, that's like everything that is like super cutting edge is expensive and usually for the privileged.
Thank you,
Yeah,
Professor.
and it also
We
it costs a lot of money to develop all this stuff. It's tens of millions of dollars, and so you know, you got to look at it that way too. Is that if they didn't charge so much for the therapies, then they wouldn't be working on them in the first place. And so you have two options. You can either just not have the therapy because nobody worked on it because it was too expensive to work on and they're never going to make their money back. Or you can have option B, where it's very expensive at first and only a few people, the privileged, get it. But then over time, it becomes cheaper and then everybody gets it. And you can't have option C where it's just cheap for everyone from the beginning, because that's capitalism. I mean, unless the government were to put more money into developing the therapies, then you could just keep it cheap the whole time. But they don't do that.
Yeah. Well, we've nearly run to the end of our hour. You know, the recent shift to be able to, you know, for a long time since the Affordable Care Act started, there was a prohibition against negotiating with Medicare on pricing, right? But now that's over. But the argument still is, if there's too much downward pressure on pricing of these amazing therapies, then that will curtail R&D, motivation for R&D. This has been a lively discussion. We've been going over the vast topic of gene editing, and I want to thank you all three, Professor Owens, Professor Segal, and Urschitz, for your great service to humankind. Roger?
That was absolutely fascinating. And it is recorded, and it will be available shortly on the Better Tomorrow Speaker Series YouTube channel, and eventually likely edited in a Hawaii Book and Music Festival website. So it can be accessed almost indefinitely. Thank you very much. It was really, I learned a huge amount, and I'm sure our attendees did as well. I have one final pitch, if you will, which is we do intend to be in person next year. It takes a lot of funding. So we're having a fundraiser on January the 26th. And I hope everyone will put that in their calendar, and I will remind them endlessly for the rest of this month and beyond.
You know, I think do you think that you guys can develop gene therapy that makes people more inclined to fund the Hawaii Book and Music Festival?
We'll work on that.
Okay, thank you so much.
Thank you very much.
Bye-bye.
Thank you. Bye.